So we're going to move on to oral presentations for 6 of our presenters. So the first person that's going to come up today is Xinjian Cen, and she will share her summer research with us. So welcome.
Okay, good afternoon everyone. Thank you so much for joining us today. I'm Xinjian Cen from the ACRS lab, and today I will be presenting about the interactions of 2 proteins, PDPN14, which is a phosphatase, and TAZ, which is a co-transcriptional factor. And both of these proteins can affect angiogenesis, which is the development of blood vessels. Imagine yourself having blood vessel malformations that cause bleeding, or Y is suffering from hereditary hemorrhagic telangiectasia, HHT, which is a genetic disorder of angiogenesis. Here are a few examples of HHT phenotypes, including a pulmonary AVM, brain AVM, as well as skin telangiectasia. In previous studies, genetic variations in PDPN14, which stands for protein tyrosine phosphatase non-receptor type 14, has shown to modify HHT phenotypes. Moreover, one of the main functions of PDPN14 is to reduce YAP-TAZ level as well as maintaining their cytoplasmic localization. In addition, YAP-TAZ has shown to affect angiogenesis, so this all suggests that there is a connection between PDPN14 and angiogenesis. Therefore, my main focus question was, does mutations in PDPN14 actually affect angiogenesis? To find out, we designed different mutants of PDPN14, which includes the PPXY mutant, a E189G mutant at the firm domain, as well as a firm domain deletion and a PTP domain deletion. Here are our experimental methods. First is molecular cloning, in which we cloned our mutant plasmids in bacteria and used them to transfect HEC293 cells. Next, we proceed to Western blot, in which the proteins are extracted and analyzed with antibodies. Lastly, we also done an immunofluorescence, in which we stain the cells with antibodies and image them under a microscope. So here is our first result. This is a Western blot of the different PDPN14 mutants. We use non-transfected cells as our negative control, as well as calnexine for our loading control. Our first finding is that the E189G mutant is making PDPN14 less stable, as we see that it's giving a fade band of PDPN14 in comparison to the others. Next, we also found that the firm domain contains PDPN14's ubiquination site. Ubiquination is just when a protein gets turned over. We see that with the deletion of the firm domain, we get a high level of PDPN14, and there seems to be no ubiquination. To confirm our findings, we look to our immunofluorescence data. Here, PDPN14 is stained green, and the nucleus is stained blue with DAPI. Here we see that PDPN14's localization is affected by the firm domain, as we see in the wild type and all the other mutants. PDPN14 tends to be cytoplasmic, whereas with the firm domain deletion, PDPN14 tends to be nuclear. Next, we also see that the E189G mutant is making PDPN14 less stable, as we see that the protein is speckled with the green color. Next, we also found that the firm domain deletion and PTP domain deletion is giving us a higher level of PDPN14, as we see that the color is much more intense. This is consistent with our previous findings in our Western plot. Another assay that we did was we combined TAZ and the different mutants of PDPN14 and overexpressed them in HEC293 cells. Here, as I mentioned before, one of PDPN14's main function is to downregulate TAZ level, and we see that in our data here. We see that with the combination of wild type and TAZ level is much lower than only TAZ getting overexpressed alone. However, with the PPXY mutant and the PTP domain deletion, we see that TAZ level remains very high. This is suggesting that PDPN14 is losing its interaction with TAZ, and these two domains are possibly the interaction sites between these two proteins. In conclusion, we found that the PBXY site and the PTP domain on PTBN14 is required for its interaction with tasks, as well as PTBN14's firm domain actually affect its level and localization. For future directions, we would like to refine the binding site of these two proteins, as well as repeating our experiments with different cell confluencies, as well as investigate in other proteins that might have interact with PTBN14. In doing so, we hope to learn more about the molecular machinery of diseases and angiogenesis in cancer development, and ultimately provide new drug targets that are related to angiogenesis diseases. And that's the end of my presentation. Does anyone have any questions, comments, or confusions? Yes?
Maybe it's a confusion. What is the significance of the localization of this protein regarding their function?
So PTBN14's localization, well, first, PTBN14 is a phosphatase, so meaning that it removes phosphate groups from proteins, so that either activates or inactivates that protein. And by entering into the nucleus, it can bring out proteins that are involved in gene transcription, so that might reduce or introduce more gene transcription.
So it's more potent.
Yeah. Is that conclusion good? Or? Okay. Does anyone else have questions or any confusion that would like me to explain? No? Okay. Well, thank you so much for listening to me, and I would like to give a special thanks to my mentor, Aunt Mamai, and Dr. Rosemary for making this possible me, and as well as the Ackers Lab. Thank you guys so much for coming. Also, I would like to thank Ms. Kilpack, John, and the LSRP committee for this amazing opportunity. This is my second year working in the Ackers Lab, and I definitely enjoy every moment I have in research. Thank you so much.